In this report, we look at the differences in the intestinal microbiome of mice following transfer between laboratories. To investigate this, 16S ribosomal RNA was obtained from mouse species for 16 animals of different age and sex. The first step of the analysis is a thorough investigation of associated metadata to help us understand the relationships between experimental variables. We have relevant information on a per mouse basis of the batch or location where the mouse came from, as well as things like sex and age. We can see that metadata variables are equally distributed across the factor of interest, which is location or batch, and there's no need to adjust for any confounders in this analysis. We next perform a robust QC of the sequencing data, looking at base quality and adapter presence in each of the sequencing files. We need to take account of anything that may negatively impact on downstream analysis, so we have trimmed low-quality ends from reads, along with any adapter contamination, prior to taxonomic classification at the genus level using Kraken and the NCBI 16S reference sequence set. First, we looked at 4 measures of alpha diversity, which is a measurement of diversity within samples, and found that there is significantly greater levels of diversity in animals post-location transfer. No differences in alpha diversity were found between mice of different sex or age within a given location. Next, we looked at beta diversity, which is a measurement of diversity between samples, and this is visualized in this principal component plot. What we can see here is a clear separation of samples by location. We have a cluster in blue on the left and a cluster in green on the right, indicating a shift in microbiome composition post-location transfer. We went on to formally test the differences in genus abundance between locations and found a significant increase in a number of genera, including Bacteroides, Monoglobus, and Dinitrobacterium, post-location transfer, and a significant decrease in a number of genera, including Adlercreaceae and Lactobacillus, amongst others, post-transfer. The findings in this report clearly have important implications for animal experiments. There is clearly a population shift following transfer between laboratories, which has resulted in a large shift in the gut microbiome in these animals. And this should be taken into account in the design and interpretation of animal experiments with a similar design.